overview of the renal physiology. So the first part basically is just anatomy. So it's just a revision on anatomy so that later on we'll go into the physiology, because for you to appreciate the physiology, you always need to remember the anatomy. So that's where we start from. So the anatomical function or the respiratory or the renal system, then later on we'll look at the actual physiology. So from this diagram, you can appreciate the two kidneys, one on the left part of the body and the other one on the right part of the body, that are connected to the urinary bladder via the ureters. So you have the two ureters that are connecting the kidneys to the urinary bladder. Then between the outside world and the urinary bladder, you have the urethra. So in the penis, or just around the vagina there, you have the urethral opening, where now urine can be exposed to the outside, or it can be voided to the outside. So this is just a simple diagram explaining the renal physiology. But the major component of renal physiology are the kidneys. Within the kidneys, there are structures which we call the kidney nephron. So the functional unit of the kidney is the kidney nephron. Once you understand how the kidney nephron operates, then you are closer to understanding renal physiology. But remember, physiology is all about mechanisms that you need to understand. If you understand those mechanisms, if a question comes in an exam, it could be an essay question, short answer question, you should be able to tackle those questions. But I should mention here that I was disappointed with the way you answered your exams, especially when it comes to essays. At least MCQs, maybe someone can guess here and there, but when it comes to essays, I was highly disappointed. And most of you managed to pass because of the labs that you submitted. So we had to allocate a lot of marks towards the labs so that you pass. Otherwise, the performance wasn't good at all. How can you fail to answer those simple questions related to blood physiology, blood typing, cross-matching? You are unable to answer those. We did the lab and we took time to explain blood physiology, but I was highly disappointed with the performance. So I hope going forward, you will improve. So especially with the essays, you need to work on your essays, because I don't expect you to give me 2 pages. If you are answering essays, I give you 3 essays or 4 essays, then somebody manages to answer just within a page those essays, and then you expect that you're going to pass physiology. It's not like that. So the marks that you have, you guys, is not a true representative of what you performed in the exam. Otherwise, if we were very strictly, most of you would have been repeating by now. Yeah, so it's not a warning, it's just an advice to say, going forward, you really need to work hard on yourself so that when you have an exam, you write something that is closer to what we are lecturing you. Many of the times we give you a chance to ask questions, you don't ask questions. Then when questions come, you don't really give us what we are looking for as lecturers. So I hope that will change. Okay. So as renal physiology, there are unit objectives that you need to understand. So these unit objectives, you can use them as a student to study, okay? So what are the unit objectives? It means that at the end of this unit, I expect you as a student to be able to describe the morphology of a typical nephron in its blood supply. So you should be able to draw a kidney nephron and also to explain the blood supply to the kidneys. Then you should be able to define autoregulation and list the major theories advanced to explain autoregulation in the kidneys. So you should be able to explain autoregulation. Why is it that the glomerular filtration rate is maintained despite changes in blood pressure? Somebody can be hypertensive or hypotensive, but the glomerular filtration rate is going to be maintained. So what autoregulation mechanisms are involved to maintain the glomerular filtration rate so that you don't have changes? Because if the glomerular filtration rate is increasing so much, sometimes you can be losing a lot of fluids via the urine. then a patient can be dehydrated. So what mechanisms are at play to maintain the glomerular filtration rate? We are talking of autoregulatory mechanisms by the kidneys themselves. So you need to understand those autoregulatory mechanisms. Then you need to define glomerular filtration rate and also describe how it can be measured. So the glomerular filtration rate, you should be able to define it. You should be able to describe how it can be measured. So there are different methods you can use to measure the glomerular filtration rate. So what are those methods that are used to measure? Then list the major factors affecting the glomerular filtration rate. So this glomerular filtration rate, what are the factors affecting it? So you should be able to list the factors and also describe how they are affecting the glomerular filtration rate. I hope I'm communicating and you're getting what I'm saying. So these are just the objectives. So it means at the end of this unit, as a student, you as an individual, you should be able to explain all this. Then you should be able to outline tubular handling of sodium and water. So you're talking of filtration of sodium and water, and then later on there will be reabsorption. So how are the kidneys handling the sodium and water in the tubes, in the kidney nephron, in short? Should be able to discuss tubular reabsorption and secretion of glucose and potassium. So this is also very important when you're talking of reabsorption of glucose. Glucose is so small that it can be filtered on the glomeruli. So the glomerulus, that is just singular. If they are a lot, they are called glomeruli. So there could be filtration of glucose, but glucose is still needed by the body. So you don't want to get rid of the glucose. So the kidneys, they are also involved in reabsorption of the glucose so that it goes back to the circulation. So which part of the kidney nephron is involved in the reabsorption of this glucose and also potassium? You should be able to describe how the countercurrent mechanism in the kidney operates to produce hypertonic or hypotonic urine. So now these countercurrent mechanisms I'm talking about, later on you notice that there are certain channels that are found within the kidney nephron that are involved in creating what we call osmotic gradient or hypertonic medium within the medulla of the kidneys. So these channels that are involved in creating the osmotic gradient, there is a mechanism which is called countercurrent mechanism that is going to facilitate the reabsorption of water. So don't worry, by now maybe you might not understand because I'm just listing what you need to understand. So you understand later on. Then you should be able to list the major classes of diuretics and how each operates to increase urine flow. So there are diuretics drugs that are used. So you need to know them and how they operate, the mode of action of these drugs. So you might cover this maybe in pharmacology, but in physiology, because physiology, we are more interested in the mechanisms. So you need also to know the mode of action of these drugs and how do they affect the function of the kidney nephron. Then last but not the least, describe the voiding reflex. So there is the voiding reflex that is called micturition or urination if you want. Then you should also be able to draw a cystometrogram. So these are just objectives. So as we are starting this topic, the renal system, I know it might be a bit confusing because we'll be alternating with Dr. Mshawati. I come in with renal physiology, he comes in with GIT. It could be quite confusing. I agree with you as a student. But I mean, as a student, you should be ready for anything, because even when you're working in those wards, in those clinics, it's not that the patients that will be coming to you have similar challenges. So there are different diseases. So you have to switch. Maybe you are dealing with a patient to do with GIT. Next minute, there's a patient to do with the renal. So you should be able to swap, your thinking. Okay. So nevertheless, whatever I'm going to lecture you, That's what I'm going to give you a test on. Whatever Dr. Mshawati is lecturing you, you also give you a test. So we're not going to mix the questions. So if I'm giving a test, it will be just the components that I've lectured. If it's Dr. Mshawati giving a test, you also give you a test based on what is going to cover. Okay. So now those were the unit objectives. But what about this lecture that you are having now, the introductory lecture? What do I expect from you? So it means at the end of this lecture, you should be able to understand the general functions of the urinary system. What are the general functions of the urinary system? Then a quick overview of the functional anatomy of the urinary system. So the anatomy, before we build on the physiology. Okay, so what are the major functions? Okay, sorry about that. I was mistaken. I was thinking that I'm supposed to cover the GIT with you guys, but we've already started with Dr. Mshawati. So we are going to switch. Let's discuss renal physiology. So renal physiology, basically you're looking at the function of the kidneys, and a normal person should have at least a pair of kidneys. So you have two kidneys. On the left and on the right part of the body, you have those kidneys. So this is just a general overview of the renal physiology. So the first part basically is just anatomy. So it's just a revision on anatomy so that later on we'll go into the physiology, because for you to appreciate the physiology, you always need to remember the anatomy. So that's where we start from. So the anatomical function or the respiratory or the renal system, then later on we'll look at the actual physiology. So from this diagram, you can appreciate the two kidneys, one on the left part of the body and the other one on the right part of the body, that are connected to the urinary bladder via the ureters. So you have the two ureters that are connecting the kidneys to the urinary bladder. Then between the outside world and the urinary bladder, you have the urethra. So in the penis, or just around the vagina there, you have the urethral opening, where now urine can be exposed to the outside, or it can be voided to the outside. So this is just a simple diagram explaining the renal physiology. But the major component of renal physiology are the kidneys. Within the kidneys, there are structures which we call the kidney nephron. So the functional unit of the kidney is the kidney nephron. Once you understand how the kidney nephron operates, then you are closer to understanding renal physiology. But remember, physiology is all about mechanisms that you need to understand. If you understand those mechanisms, if a question comes in an exam, it could be an essay question, short answer question, should be able to tackle those questions. But I should mention here that I was disappointed with the way you answered your exams, especially when it comes to essays. At least MCQs, maybe someone can guess here and there, but when it comes to essays, I was highly disappointed. And most of you managed to pass because of the labs that you submitted. So we had to allocate a lot of marks towards the labs so that you pass. Otherwise, the performance wasn't good at all. How can you fail to answer those simple questions related to blood physiology, blood typing, cross-matching? You are unable to answer those. We did the lab and we took time to explain blood physiology, but I was highly disappointed with the performance. So I hope going forward, you will improve. So especially with the essays, you need to work on your essays, because I don't expect you to give me 2 pages. If you are answering essays, I give you 3 essays or 4 essays, then somebody manages to answer just within a page those essays, and then you expect that you're going to pass physiology. It's not like that. So the marks that you have, you guys, is not a true representative of what you performed in the exam. Otherwise, if we were very strictly, most of you would have been repeating by now. Yeah, so it's not a warning, it's just an advice to say, going forward, you really need to work hard on yourself so that when you have an exam, you write something that is closer to what we are lecturing you. Many of the times we give you a chance to ask questions, you don't ask questions. Then when questions come, you don't really give us what we are looking for as lecturers. So I hope that will change. Okay. So as renal physiology, there are unit objectives that you need to understand. So these unit objectives, you can use them as a student to study. Okay. So what are the unit objectives? It means that at the end of this unit, I expect you as a student to be able to describe the morphology of a typical nephron in its blood supply. So you should be able to draw a kidney nephron and also to explain the blood supply to the kidneys. Then you should be able to define autoregulation and list the major theories advanced to explain autoregulation in the kidneys. So you should be able to explain autoregulation. Why is it that the glomerular filtration rate is maintained despite changes in blood pressure? Somebody can be hypertensive or hypotensive, but the glomerular filtration rate is going to be maintained. So what autoregulation mechanisms are involved to maintain the glomerular filtration rate so that you don't have changes? Because if the glomerular filtration rate is increasing so much, sometimes you can be losing a lot of fluids via the urine. Then a patient can be dehydrated. So what mechanisms are at play to maintain the glomerular filtration rate? We are talking of autoregulatory mechanisms by the kidneys themselves. So you need to understand those autoregulatory mechanisms. Then you need to define glomerular filtration rate despite and also describe how it can be measured. So the glomerular filtration rate, you should be able to define it. You should be able to describe how it can be measured. So there are different methods you can use to measure the glomerular filtration rate. So what are those methods that are used to measure? Then list the major factors affecting the glomerular filtration rate. So this glomerular filtration rate, what are the factors affecting it? So you should be able to list the factors and also describe how they are affecting the glomerular filtration rate. I hope I'm communicating and you're getting what I'm saying. So these are just the objectives. So it means at the end of this unit, as a student, you as an individual, you should be able to explain all this. Then you should be able to outline tubular handling of sodium and water. So you're talking of filtration of sodium and water, and then later on there will be reabsorption. So how are the kidneys handling the sodium and water in the tubes, in the kidney nephron, in short? Should be able to discuss tubular reabsorption and secretion of glucose and potassium. So this is also very important when you're talking of reabsorption of glucose. Glucose is so small that it can be filtered on the glomeruli. So the glomerulus, that is just singular. If they are a lot, they are called glomeruli. So there could be filtration of glucose, but glucose is still needed by the body. So you don't want to get rid of the glucose. So the kidneys, they are also involved in reabsorption of the glucose so that it goes back to the circulation. So which part of the kidney nephron is involved in the reabsorption of this glucose and also potassium? You should be able to describe how the countercurrent mechanism in the kidney operates to produce hypertonic or hypotonic urine. So now these countercurrent mechanisms I'm talking about, later on, you notice that there are certain channels that are found within the kidney nephron that are involved in creating what we call osmotic gradient or hypertonic medium within the medulla of the kidneys. So these channels that are involved in creating the osmotic gradient, there is a mechanism which is called countercurrent mechanism that is going to facilitate the reabsorption of water. So don't worry, by now maybe you might not understand because I'm just listing what you need to understand. So you understand later on. Then you should be able to list the major classes of diuretics and how each operates to increase urine flow. So there are diuretics drugs that are used. So you need to know them and how they operate, the mode of action of these drugs. So you might cover this maybe in pharmacology, but in physiology, because physiology, we are more interested in the mechanisms. So you need also to know the mode of action of these drugs and how do they affect the function of the kidney nephron. Then last but not the least, describe the voiding reflex. So there is the voiding reflex that is called micturition or urination if you want. Then you should also be able to draw a cystometrogram. So these are just objectives. So as we are starting this topic, the renal system, I know it might be a bit confusing because we'll be alternating with Dr. Mshavati. I come in with renal physiology, he comes in with GIT. It could be quite confusing. I agree with you as a student. But I mean, as a student, you should be ready for anything, because even when you're working in those wards, in those clinics, it's not that the patients that will be coming to you have similar challenges. So there are different diseases. So you have to switch. Maybe you are dealing with a patient to do with GIT. Next minute, there's a patient to do with the renal. So you should be able to swap, your thinking. Okay. So nevertheless, whatever I'm going to lecture you, that's what I'm going to give you a test on. Whatever Dr. Mshavati is lecturing you, you also give you a test. So we're not going to mix the questions. So if I'm giving a test, it will be just the components that I've lectured. If it's Dr. Mshavati giving a test, you also give you a test based on what he's going to cover. Okay. So now those were the unit objectives. But what about this lecture that you are having now, the introductory lecture? What do I expect from you? So it means at the end of this lecture, you should be able to understand the general functions of the urinary system. What are the general functions of the urinary system? Then a quick overview of the functional anatomy of the urinary system. So the anatomy, before we build on the physiology. Okay, so what are the major functions of the kidneys? Major functions of the kidneys. So even before we tap into the information, already in your mind, you have some answers. They could not be so accurate, but at least you know the function of the kidneys. So maybe before we proceed, can you just mention some of the functions of the kidneys? What are the functions? Anyone?
Excretion of urine.
Yeah, excretion of urine, that's correct. What else? Remember, with me, I told you to say, I like my lectures to be interactive. I would have loved that we have these lectures face to face, but where we are here, it's very far from Kawe, so we have no choice there but just to go online. But I always want my lectures to be interactive. So you as a student, sorry, I'm not getting that. I wasn't getting what you were trying to explain. So somebody is saying regulation of blood pressure. Yeah.
Production of hormones like erythropoietin.
Excellent. So production of hormones like erythropoietin. What other hormones are being produced by the kidneys? So there's the production of renin, prostaglandins. The kidneys, they are also producing that. And also remember the conversion of the active form of vitamin D, as the vitamin D is coming from the liver, it's coming in form of 25 hydrocholecalciferol. When it goes to the kidney, there is a final conversion with the presence of parathyroid hormone. So it will be converted into 125 dihydrocholecalciferol. That is also taking place within the kidney. So those are the hormones. What else?
So what about removal of toxic substances such as urea?
Yeah. So excretion of waste products that are coming from metabolism. What else? Maintenance of fluid balance. Yes. So some people are just typing, which is not bad, but I would like that you speak out so that other people can also benefit from your contributions. Because when you type, not everyone is checking the chat, so they might miss your point. So yeah, maintenance of fluid balance. I agree with you. What else?
Gluconeogenesis.
Gluconeogenesis. Yeah, there are some cells that are undergoing that process, but not many. Gluconeogenesis mainly is taking place within the muscles and the liver cells, the hepatocytes. Okay, so I know you have some ideas. So let's look at the major function. So the first major function of the kidneys, it's regulation of different things. So what does it regulate? So the kidneys, they are involved in the regulation of body fluid osmolality and volume. So the regulation of osmolality and volume. Remember, osmolality is the number of osmoles per kg or per liter of solution. The concentration of these solutes in solution. So those concentration of solutes in solution is controlled by the kidneys because the kidneys are the ones that are involved in secretion. So if a particular ion or electrolyte has increased in circulation, you find that the kidneys will excrete much of it. So it's going to control the osmolality of the fluids, and at the same time, is also controlling the volume. What do I mean? If right now you take a break and then you drink a lot of water, maybe you drink a liter of water, in the next maybe 30, 40 minutes, you have this urge to go and urinate. Why? It's because the kidneys, they are involved in control of volume. So as you are taking that liter of water, your blood volume is going to increase. If the blood volume is increasing, and also the glomerular filtration rate is going to increase so that you filter much of this fluid, then you will now excrete the fluid in form of urine. So it's also involved in the regulation of volume. Regulation of electrolyte balance. So those electrolytes, so you have dissolved solutes in water that are also controlled by the kidneys. Remember, the kidneys, they are involved in filtration, secretion, and reabsorption. So those three processes of the kidneys, the filtration, secretion, and reabsorption, can bring about balance or regulation of these electrolytes. Acid-base balance, the kidneys themselves, they are involved in secretion of hydrogen ions. So in case of acidosis, you know to say, if a patient is having acidosis, there is an increase in the hydrogen ions, and the pH is simply the negative logarithm of the hydrogen ions. So if the hydrogen ions are increasing in the body, in circulation, the pH will be going down. So how do the kidneys come in as a compensatory mechanism to correct the pH or to maintain the pH? So those extra hydrogen ions can be excreted by the kidneys. Once the hydrogen ions are excreted in the urine, you know to say that the pH now will start going towards the normal. So it's involved in acid-base balance. So if there are too much acids in the body, the kidneys can excrete some of it. If there are too much bases in the body to the extent that your body pH is becoming more alkaline, you find that the kidneys can also be involved in excreting some of these bases like bicarbonates and other issues. So it's involved in the regulation of acid-base balance. Blood pressure, somebody mentioned that. So the kidneys, they're involved in regulation of blood pressure. How is it involved? When there's an increase in blood pressure, there will be an increase in the renal plasma flow and also renal blood flow. If there's an increase in renal plasma flow and renal blood flow, you know to say that the glomerular filtration rate is going to increase, and then you'll be filtering much of the plasma so that you produce a lot of urine. So if you are producing a lot of urine, it means your plasma volume will start reducing. if the plasma volume is reducing, your blood pressure is going to decrease. And at the same time, if your blood pressure is reducing, the kidneys, they are also involved because they can produce certain hormones like renin, and you know the function of renin also. You know to say that this renin is produced by special types of cells that are found within the juxtaglomerular apparatus. In the juxtaglomerular apparatus, there are different types of cells there. There are granular cells that are responsible for the production of renin. We have the macular denser cells that are responsible to sense the presence of sodium. So they are sodium sensitive cells that are found in the proximal, not proximal, but in the distal convoluted tubules. Then on top of that, there are also what we call mesangial cells. So when blood pressure is reducing, the macular denser cells will sense the presence of sodium chloride. Then they can stimulate the cells that can produce renin. And this renin, what does it do? Renin is more like a hormone or an enzyme that can catalyze a reaction to convert angiotensinogen that is produced by the liver into angiotensin 1. So once renin is released, it will go into circulation, then it will facilitate the conversion of angiotensinogen into angiotensin 1. This angiotensin 1 will be transported to the lungs. Within the lungs, we have the ACE, which are called angiotensin converting enzymes that will convert angiotensin 1 into angiotensin 2. The angiotensin 2 has got a lot of functions. So it can go to the blood cells, to the red blood cells, not necessarily red blood cells, but the smooth muscle cells to cause vasoconstriction of smooth muscle cells. Then at the same time, you know to say that angiotensin 2 go and stimulate the adrenal gland to release a lot of aldosterone. So the angiotensin 2 can go and stimulate the adrenal gland to produce aldosterone. What is the function of aldosterone? It will go back to the kidneys. Then within the kidneys, it will go to the nephron, the kidney nephron. It will facilitate reabsorption of sodium. So if there's reabsorption of sodium, even water reabsorption will be facilitated. So if you are reabsorbing much of sodium and water from the kidneys, you'll be producing less urine, but the plasma volume will start increasing. Then the blood pressure will increase. So the kidneys, they are also involved in blood pressure regulation. Okay. So that was the first major function. It's involved in regulation of a lot of things that are listed there, and I've tried to explain that. Then the kidneys, they are also involved in excretion of metabolic wastes. So excretion, you're talking of getting rid of these metabolic products. So the metabolic products, these are waste products that are coming from metabolism. So if these waste products are building up within the body or they are accumulating within the body, they can be toxic to the body cells. So they need to be excreted. So the kidneys are the ones that are involved in excretion of these waste products. They could be ammonia. So there are a lot of waste products that are coming from the cells that needs to be excreted. On top of that, there is excretion of foreign substances like pesticides or chemicals. For instance, if you are sick, you're going to take medicines. Those medicines are foreign substances to the body. So after they do their work, they need to be excreted. So the kidneys, they are involved in excretion of foreign substances like drugs, okay? Then they are also involved in excretion of excess substances like water. So if you have edema, for instance, you find that the kidneys, they are involved in excretion of water or other excess substances that can accumulate within the body. The other major function is secretion. So what does the kidney secrete? So there is production and secretion of these substances. So mainly here is secretion of hormones. So the hormones that are secreted by the kidneys, somebody mentioned erythropoietin. So there is production of erythropoietin. At the same time, there is also production of thrombopoietin. The kidneys can also produce a bit of thrombopoietin. So there is production of erythropoietin and thrombopoietin. Then there's also secretion of the active form of vitamin D, which is 125 dihydrocholecalciferol, or the vitamin D3, the active form, the activated vitamin D3. Then there's production of renal. There are also other hormones that I mentioned, like prostaglandins, that are also produced by the kidneys. So these are the major functions of the kidneys. So you can see just from this slide, there are just few information here. But if I ask you an essay to explain the major functions of the kidneys, you're not just going to say regulation, excretion, and secretion. You need to explain those points. Okay. Are we all on the same page?
Yes.
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 Yeah. Okay, so we proceed. So that was just the general function of the renal system. But now let's just have an overview of the functional anatomy of the renal system. So the urinary system consists of the kidneys, like I said. So you can see the kidneys. From there, you have the ureters. From the ureters, we have the urinary bladder. From the urinary bladder, you have the urethra. The urethra is going to open at the glans penis or within the vulva of the vagina. There is an opening, the urethral opening, just near the clitoris there. You have urethral opening that is involved in getting rid of the urine, okay, in a process which is called micturition. So this is just the general overview of the urinary and the renal system, not urinary system, the renal system. So the kidneys are the functional unit of the system. So the major functional unit of the renal of the urinary system are the kidneys themselves. Then we have the ureter, the bladder, and the urethra. These are conducting or storage components. So they are involved in conduction of urine or transportation of urine and storage of urine. So they are called conducting storage components of the renal system. The location of the kidneys. So the kidneys, they are located to they are lateral to the vertebral column, high on the body wall. So at the back of the person, near the vertebral column, that's where you have the kidneys. So they are under the floating lips. So the floating lips, they go all the way, starting from the front, and then they go all the way behind, like that. So under these floating lips, that's where you find the kidneys, but they are in the retroperitoneum. What do I mean when I say retroperitoneum? Remember, you have the peritoneal cavity where you have the visceral organs, especially the intestines, you know, the stomach itself. It's found within the peritoneal cavity or the abdominal cavity. So this cavity is enclosed within a membrane which is called the peritoneum membrane. But the kidneys, they are not actually located inside the peritoneal cavity. They are outside. That's why their position is retroperitoneal. So if you are to do a surgery on a patient and then you want to access the kidneys, you're not going to approach them from the front of the patient, because if you open the abdominal cavity, what you're going to see under there are intestines. You need to remove the intestines. You won't still see the kidneys because they are behind the peritoneum. So their location is retroperitoneum. So the best way you can access the kidneys is by approaching the patient from the back. From the back here, you just need to cut the muscles and then you'll be able to see the kidneys that are covered in a capsule. On average, the kidneys are heavier in males as compared to females. In males, they can weigh about 150 grams. In females, they can weigh about 135 grams. So why are the kidneys heavier in men as compared to females? You know already that the volume of blood is more in men as compared to females. So you need to filter the blood. Okay, so this is where we ended. I was explaining the weight of the kidneys in females and in males. So always remember that in males is 135, females is 150. So these figures, by now you understand, MCQs. I like bringing the figures because these are facts that you either you know or you don't know. So I can bring a question in MCQs about the weight of the kidneys. So if you can't remember, then it becomes an issue. So the dimensions of the kidneys, so in terms of the length is about 12 centimeters. Then, I mean, the height is about 12 centimeters, and then the length is about 6 centimeters. Then the width or the thickness is about 3 centimeters. This is just a 3D picture of the kidney. The position of the kidneys I've already explained. So the surface anatomy of the kidney, what covers the kidney, the location we've already said that is retroperitoneal, so it's behind the peritoneum. So it's not among the visceral organs that are found within the peritoneal cavity. I hope you understand that. Then in terms of the size of the kidney, it's just like a bar of soap. It's bean shaped, like you have seen. Then you have the hilus indentation, where you have the introduction of the innervation and also blood supply. So they go through the hilus. So you can see in this diagram that you have this indentation, the hilus, and then you can see blood supply to the kidneys and also innervation to the kidneys. The coverings of the kidneys, the outermost, you have the renal fascia. So the renal fascia is just a fibrous tunic. So this is just a hard connective tissue material that covers the outside. Then under the fibrous tunic, you have adipose tissue that will provide comfort to the kidneys, or it will provide structural support so that the kidneys cannot be easily damaged by the surrounding tissues. There is this layer of adipose tissue. So it's going to protect the kidney. Then after the adipose tissue, you have the renal corpuscle or capsule. The renal capsule is the membrane that is covering the outermost of the kidney structure itself. So the kidney itself is covered in a capsule, which is called renal capsule. And this renal capsule, it anchors the kidney to the body wall, and it's continuous with the peritoneum. So it's going to provide structural support so that when you're standing up, the kidneys don't fall down. Because remember, they are up there just under the floating lids. So when you're in an upright position, the kidneys won't drop towards the anus. Why? It's because they are secured by the renal capsule and also the renal fascia that is attaching to the surrounding structures. Okay, but when you compare the two kidneys, which one is a bit down as compared to, if you compare the right kidney and the left kidney? There is one which is more superior, the other one is more inferior. So which one is more superior, which one is superior? Inferior, I mean. I hope you've gotten my question. So each and every person, they have two kidneys. There is one kidney which is more superior to the other kidney. So which one is more superior to the other kidney in terms of the position in an upright standing person?
The left one is superior to the right.
The left one is
Superior to the right.
okay. So do we all agree with what she's saying? She's saying the left one is superior to the right kidney. Do we agree with that? So from that diagram, you can tell the answers are there. So the left is more superior. The right kidney is inferior. Why is that? You can explain why it's like that. Anyone? The in-service. You know about this information. Let's share knowledge. Don't just be quiet. So why is it that the left kidney is more superior to the right one? Okay, so it appears that we both don't know, so let's just proceed. Even me, I don't think I know. So since no one is willing to share that information, we'll leave it like that and proceed. Okay, so now let's look at the overview of the functional anatomy. So this is now the actual kidney. The coverings of the kidney we've discussed. You have the renal fascia, the adipose tissue.
That's what to make it dry, sir.
Yes, please go ahead.
Yes, it could be because of the liver on the right side.
So was that difficult to mention, please? I know you guys, you know this information. that the liver is more on the right side than the left side. So if the liver is more on the right side, it's going to push the kidney somehow down, not so. And if you can't really remember that, the best answer you could say is just that you just have to mention to say, that's how God designed it, that the left kidney should be superior to the right one. It's about creation. So that's how he positioned it when he was creating a human being. That is if you don't really know the physiology or the anatomy, but just with general knowledge, you can, using your logic, you can explain to say the kidney is more on the right side. So if it's that, so you find that the kidney, the liver is more on the right side, so it will push the kidney down. Okay, so we proceed. So this is the actual kidney. Now looking at the cross section of the kidney, when you cut the kidney a cross section, what structures are you going to appreciate? So it's divided into the outer cortex. So you can see from this diagram, you have the outer cortex. So this outer cortex is highlighted in yellow in this diagram. You've seen the cortex? So this is the cortex, the outermost part of the kidney. It's called the cortex. So just under the capsule, the kidney capsule, you have the cortex. If you peel the capsule, what you're going to see is the cortex of the kidney. Then inside, after the cortex, you have the inner medulla. So you have the inner medulla. So the medulla is the inner part of the kidney. And at the center, you have calyces, the minor calyces, the major calyces. Then you have the pelvis that will join to form the ureter. Okay, the renal pelvis will join to form the ureter. So the functional unit of the kidney is the nephron. In this diagram, you can appreciate the nephrons, the two nephrons that are drawn there on this part of the kidney. So you have two different types of nephrons that we are going to appreciate later on. You have the cortical nephron. So the cortical nephron, the loop of Henle doesn't really descend deep into the medulla. So the majority of this nephron is found within the cortex. That's why it's called the cortical nephron. Then you have another nephron by which the glomerulus is at the junction between the cortex and the medulla, or the medulla, if you like. So that nephron is called the juxtamedullary nephron. You can see it in here as well. So there are two different types of kidney nephron that we'll explain later on. Okay, so you can see the nephron there that are highlighted in green. The renal pelvis, and then you have the ureter. Okay, so still looking at the cross section of the kidneys, other internal structures that I haven't mentioned here. So we start. So we have the pyramids. So in the medulla or the medulla, you can appreciate the pyramids. So these more like triangular shaped structures are called pyramids. So it's more like forming a pyramid. You can see that. So near the cortex, they are broad. As you are going inside the medulla, they will narrow down to form the papillae. So these are called renal pyramids. So they are pyramids. Towards the end, you have the renal papillae, and the renal papillae are the ones that will open into the minor calyces. Then minor calyces will join to form the major calyces, and then they will join to form the renal pelvis, and then the renal pelvis will continue as a ureter. So let's start. So you have the pyramids, the renal pyramids that are opening into the papillae. So you have these projections of the pyramids that are projecting or protruding into the minor calyces. So these are called the papillae. So this is where you find the opening of the collecting ducts. So later on, you'll see that the kidney nephron is more like a tube that is connecting to the collecting ducts, and the collecting ducts are opening into the renal papillae. So these renal papillae, they're more like projections or protrusion into the minor calyces. So from the renal papillae, you have what we call minor calyces, and the minor calyces will join. So you can see you have minor calyces, there's 1 here, 2, 3, that are joining to form a major calyx. So you can see they are forming major calyces, and the major calyces are going to join to form the renal pelvis. The renal pelvis, and this renal pelvis will continue as the ureter. You can see it down here. After the renal pelvis, you have the ureter that is connecting the kidneys to the urinary bladder. So this is more of anatomy. So this is a kidney nephron. So you can see the nephron, the different parts of the nephron. You have the glomerulus, the Bowman's capsule with the Bowman's space. Then you have the proximal convoluted tubule. You have the loop of Henle. Then the distal convoluted tubule that is connecting to the collecting tubules. And the collecting tubules will connect to form the collecting ducts. The collecting ducts are the ones that are opening into the renal papillae, that are projecting into the minor calyces, then to the major calyces, to the renal pelvis, then all the way via the ureter to the urinary bladder. Okay, so this is more of anatomy. Then you need to realize to say that associated with these tubes or the kidney nephron, we have the adjacent to them, we have capillaries, and those capillaries that are adjacent to the tubes are called peritubular capillaries. So the peritubular capillaries are associated with the proximal and distal convoluted tubules. But in the loop of Henle, they changed their name. So the name of the peritubular capillaries in the loop of Henle, they are called vasa recta. So vasa recta are just simply a fancy name for the peritubular capillaries that are found associated with the loop of Henle. So this is the kidney nephron, and these kidney nephron, mainly they are involved in reabsorption, secretion, filtration. Those are the major functions that we'll be explaining later on. So the nephron consists of the vesicular components. So the vesicular components, you're talking of blood vessels. So there's innervation, there's also blood supply to the kidneys. So the blood supply, that is the vesicular components of the kidney nephron. So you have the one that is bringing in blood is called the afferent, starting with A, afferent arterioles. So A simply means approaching the glomerulus. So they are called afferent. So the one that brings blood is called afferent, starting with A, and the one that will take away blood from the glomerulus, they are called efferent arterioles. So afferent will bring the blood, the efferent will take the blood that hasn't been filtered by the glomerulus. Okay, then later on you have the peritubular capillaries and the vasa recta. So you have the afferent and the efferent arterioles. I've already explained the glomerulus. This is more like a cup-like structure that is involved in filtration. The peritubular capillaries, they are just besides the renal tubules. So they are besides the proximal and distal convoluted tubule. Vasa recta, when you're approaching the loop of Henle, so even these peritubular capillaries will form like a loop. So that loop that is formed by the peritubular capillaries, they are called vasa recta. In other way, the vasa recta are peritubular capillaries on the loop of Henle. Then we have the tubular components, and the tubular components, these are the components that I've already explained. To say you're starting with the proximal convoluted tubule, then the loop of Henle. So the loop of Henle is divided into descending and ascending. So the descending, you have the thick part and the thin part. So you have the thick descending loop of Henle, and then you have the thin descending loop of Henle. Then it will form like a hairpin-like structure, that is forming a loop, and then it will start ascending. So as it's ascending, it will start with a thin ascending loop of Henle and a thick ascending loop of Henle that is connected to the distal convoluted tubule. And these distal convoluted tubule, they are connecting to the collecting tubules, and the collecting tubules are connecting to form the collecting ducts. So you know that pathway. The proximal convoluted tubule, the distal convoluted tubule, and also the nephron loop or the loop of Henle. These are the tubular components of the kidney nephron. Then the collecting ducts as well. And then we have tubovascular components. So it's like a tube in association with the vascular system. So the tubes of the nephron in association with the blood supply or the vascular system. It's called tubovascular component. Have you discussed renal physiology in anatomy? Not necessarily renal physiology, I mean renal anatomy.
No.
Okay, but you've heard of juxtaglomerular apparatus. Juxtaglomerular apparatus. So we have the juxtaglomerular apparatus, which is tubovascular component of the nephron. So it's partly a tube and it's partly vascular system. So the tube part of it, there are cells that are contributing to the juxtaglomerular apparatus, which are called the macular denser cells. And then you have the vascular component, which are smooth muscle cells that are involved in vasoconstriction. So I will explain later on. This is just the anatomy. So this is a simplified function of the nephron. So there are basically 4 major functions of the kidney nephron. The first one is filtration of the blood. Then there will be reabsorption and secretion. Then later on, there will be excretion. So this is just a simple or simplified function of the kidney nephron. So blood is brought in by the afferent arteriole. Then this blood is filtered. About 20% of this blood is being filtered here. So you have the ultrafiltrate. Then the 80% of this blood will just proceed to the efferent arterioles. The filtrate will start moving in the tubule. So the proximal convoluted tubule, there are channels that are involved in reabsorption. So some of the substances that are found in this fluid that are moving on the lumen of the tubule, they are going to be reabsorbed back to the cardiovascular system into the peritubular capillaries that you can see there. Then there are also other substances that were not filtered here in the glomerulus that needs to be excreted. So those substances will be secreted by the tubular capillaries back into the lumen of the kidney nephron. So you can see secretion taking place there. Then the overall thing now, you have excretion. So after reabsorption and secretion, what will remain in the lumen of the kidney nephron now will be excreted in the urine. So it will go to the bladder and to the external environment. So it will be excreted. Okay, so amount filtered, which is F, minus the amount reabsorbed, plus the amount secreted should give you the amount of the solutes that are being excreted. So this is just a simple equation. So the amount of substances that are being excreted is given by the amount that is being filtered here. So once it's filtered here, minus the amount that is being reabsorbed because it's going back to the circulation. Then plus the amount that is being secreted, if any, then it will give you the overall amount of the solutes that are being excreted. Is it difficult to understand that equation? Okay, I think everyone has understood that equation, so we proceed. This is more like a cartoon that is trying to explain the same function of the kidney nephron. So blood is brought in at the glomerulus where filtration is taking place. So after filtration, there are certain things that are being filtered which are still needed by the body. So if they are not reabsorbed, then they will be excreted. But because the body has realized to say, I still need amino acids, I still need glucose, there are pumps or channels that are involved in reabsorption. So those things that are still needed by the body, they will be reabsorbed in the proximal convoluted tubule. At the same time, there are certain things that were not being filtered here. They proceed into the efferent arteriole. When it reaches the peritubular capillaries, the body realizes to say, I should have filtered this. So what does it do? It's going to secrete it into the lumen of the kidney nephron so that it can be excreted later on. So in the proximal convoluted tubule and distal convoluted tubule, there is absorption and reabsorption taking place there. So they will modify the kind of urine that is being produced. So these factors, the reabsorption and secretion, they will modify the type of urine that is being produced depending on the requirements of the body. In the loop of Henle, this is where the countercurrent mechanism come in. The countercurrent mechanism come in because you have salt pumps or electrolyte pumps. So these pumps, they are involved in active transportation or active transport. They are going to pump. So in the thin ascending limb, so you can see you have the thin descending and the thin ascending. So in the thin ascending, you have these pumps that are pumping salts like sodium, chloride, potassium. So it's going to pump into the medulla of the kidney. The concentration of these solutes are going to increase in the medulla. If the concentration is increasing, then water will start following salt via osmosis. So in the thin descending now, water can move from the lumen into the interstitial spaces. Then later on, it will be absorbed by the peritubular capillaries. It goes back to the circulation. So this is the countercurrent mechanism that I'll explain later on. I'm just introducing. So this is just a general function of the kidney nephron. Then from the distal convoluted tubule, you have the collecting tubules that are connecting to form the collecting ducts. And you can see urine, the final urine that is being produced here after modification by secretion and reabsorption in the kidney nephron. Filtration there, reabsorption and secretion, like I've already explained. Then later on, urine will be released. So section 1, characteristics of renal structure and function. The physiological anatomy of the kidney. So there are two different types of the kidney that I'm going to explain to you. So you can see the functional unit of the kidney, we said is a nephron, like we have already explained. Then it's made up of about 1 million. So each kidney in the body, it will have about 1 million nephron. So 1 million on one side, 1 million on the other side. The total number of kidney nephron a person would have should be somewhere around 2 million nephrons. That's a lot of kidney nephrons. They are all involved in filtration. Okay. So each nephron has two major components. So you have the glomerulus and the long tube. So these are the distal loop of Henle, the proximal convoluted tubules, like I've already explained to you guys. Okay, so coming to two different types of kidney nephron. So you have a kidney nephron that is mainly located within the cortex. So this is the cortex shown in gray. Then down here, dark gray, this is the medulla. So this nephron, which is mainly in the cortex, is called cortical nephron. You can see there. The one that is also projecting mainly into the medulla is called juxtamedullary nephron. So these are the two different types of nephron. But the glomeruli of the both nephrons are found within the cortex. Why is it that the glomeruli are found within the cortex? It's because the blood supply to the cortex is more as compared to the blood supply to the medulla. So if blood supply is more to the cortex, filtration is taking place more within the cortex. So the glomeruli of both the cortical and juxtamedullary nephrons, they are found within the cortex. And then the loop of Henle are the ones that will project into the medulla, like you've already seen here. Okay. So this other diagram, I think it's just explaining almost the same things. So let's just proceed. So the two different types of kidney nephron I've already explained. You have the cortical nephron, the glomeruli in the outer cortex, and short loops of Henle that extend only short distance into the medulla. So there's only short loops of Henle that are extending short distance into the medulla. Then you have the juxtamedullary nephron, where the glomeruli are in the inner part of the cortex, and long loops of Henle which extend deeply into the medulla. So the loop of Henle deeply extend into medulla, then this is the juxtamedullary nephron. So these are the two different types. But the peritubular capillaries around the loop of Henle, they are called vasculacta, like I've already explained. This is the renal corpuscle. So the renal corpuscle is composed of a membrane that is involved in filtration. So it's composed of glomerulus and the Bowman's capsule. This is called the renal corpuscle, the structure that is mainly involved in filtration. So the membrane barrier in the filtration membrane, we have two major cells and the basement membrane. So within the glomerular capillaries, you have the endothelial cells, which is just a single layer of cells. In between these cells, you have the openings, which are called fenestrations. So the fenestrations, the pores or the opening in these capillaries, they are greater when you compare it to the openings of the capillaries elsewhere within the body. So they are called fenestrations. So the fenestrations, they are going to facilitate filtration. After the endothelial cells, they are just sitting on the basement membrane. And this basement membrane, after the basement membrane, you have the podocytes. The podocytes are the structure of the capsule, but they are covering the capillaries. So the podocytes are cells that sends the foot-like projections to cover the capillaries. But these foot-like projections, they have openings in between them. They have slits, which are called filtration slits. So for substances to be filtered, they need to cross the fenestrations of the endothelial cells in the capillaries. They need to cross the basement membrane. Then they need to pass through the slits of the podocytes to go into the Bowman space. So they will find themselves into the Bowman space. Then they will be collected by the proximal convoluted tubule that is also involved in secretion and reabsorption. So this is called the renal corpuscle. The structure of the kidney nephron, I think I've already explained. The juxtaglomerular apparatus. The juxtaglomerular apparatus, it will include the macula densa, the extraglomerular mesangial cells, and the juxtaglomerular granular cells. The granular cells are the ones that are responsible for the production of renin. The macula densa cells are the ones that are sensing sodium chloride that is moving within the distal convoluted tubule. Then we have the mesangial cells. The mesangial cells are extraglomerular, meaning that they are found outside the glomerulus. That's why they are called extraglomerular mesangial cells, which are simply modified smooth muscle cells that are involved in contraction and relaxation. So if there is contraction of the mesangial cells and smooth muscle cells, it will bring about vasoconstriction of the afferent arteriole or the efferent arterioles. That will have an effect also on the glomerular filtration rate. Okay, so you can see that within the distal tubule, you have the macula densa cells, which are modified epithelium cells. So these modified epithelium cells are the ones that are sensing sodium and chloride as it's passing within the lumen here. Then it can stimulate these cells to contract, the mesangial cells and the smooth muscle cells to contract. Then they can also stimulate the production of renin from the granular cells. So they can stimulate the granular cells to produce renin. So this structure is partly the tube and the vascular system. That's why it's called tubulovascular component of the kidney nephron. the juxtaglomerular apparatus. Are there questions so far? Am I communicating?
Yes.
Are there questions?
No.
Questions?
No, Doc.
Okay. Now let's look at blood supply to the kidneys. Blood supply to the kidneys. So remember, you have major vessels that are coming out of the heart. Those vessels, you have the pulmonary trunk that is carrying blood to the lungs. So it's called the pulmonary trunk that will bifurcate into the right and the left pulmonary arteries. Then you have the aorta itself. The aorta that will descend down. So you have the descending aorta. So as it's coming from the heart, it's ascending, then it will curve to start descending. Then it becomes the descending aorta. So as it's passing different structures of the body, the name of this descending aorta changes. So if it's in the thoracic cavity, it's called the thoracic aorta. As it goes down to pierce the diaphragm, and then it will go into the abdominal, and then it becomes abdominal aorta. It will proceed down like that. That's anatomy. So from this abdominal aorta, there are branches of renal arteries. So you can see from this descending abdominal aorta, there are branches of the arteries which will form the right and the left renal arteries. So you have the right and left renal arteries that are supplying the blood, the kidneys. So here they are. So you have these renal arteries, which are a branch of abdominal aorta or descending abdominal aorta. The major renal artery, as it's going via the hilus, they are going to bifurcate to form the segmental arteries. So you can see they will divide to form the segmental arteries. The segmental arteries, as they are approaching the pyramids of the kidney, they are going to divide to form the interlobar arteries. So you can see the interlobar arteries, they are in between the pyramids. So because they are between the pyramids, sometimes they are also called the interpyramidal arteries or interlobular arteries if you want. So these pyramids, sometimes they are referred to as lobes. So these are called interlobar arteries, which are branches of segmental arteries. Segmental arteries are branches of the renal artery. So I hope you are getting that. The renal artery is going to bifurcate or to divide into the segmental arteries. The segmental arteries, they are going to divide to form interlobar arteries. The interlobar arteries, they can communicate within the junction between the cortex and the medulla. So at the junction between the cortex and the medulla, you have the communicating arteries. These communicating arteries, they are called arcuate arteries. So you can see down there, the arcuate arteries, they are communicating the interlobar arteries. From these arcuate arteries, you have branches that are coming off there. So these branches are called interlobular arteries. These are called interlobular arteries. The interlobular arteries are the ones that are going to divide into the afferent arteries or arterioles. Afferent arterioles. The afferent arterioles, they will supply the glomerulus. From there, they will continue as the efferent arterioles. They will divide to form peritubular capillaries. Then they will join to form the interlobular veins. The interlobular veins will join to form arcuate veins. The arcuate veins will join to form the interlobar veins. The interlobar veins will join to form segmental veins. The segmental veins, they will join to form renal veins. And then this will join the inferior vena cava to take blood back to the heart. So this is the blood supply of the kidneys. So the information is there. Okay, so maybe you were not understanding what I was trying to explain. Maybe this will help. This is simply just looking at blood supply to the kidneys. So you have the renal arteries, which are branches of abdominal artery. They will divide to form segmental arteries, and the segmental arteries, they will divide to form the interlobar arteries. The interlobar arteries, they can communicate with each other via the arcuate arteries. So they are able to communicate with each other via the arcuate arteries. Then the arcuate arteries will give branches, which are called interlobular arteries. The interlobular arteries, sometimes they are referred to as radial arteries. Then these will form the afferent arterioles. The afferent arterioles, they are the ones that will now divide to form the glomerular capillaries that are involved in filtration at the glomerulus. They will continue as the efferent arterioles. Okay, so we proceed. We are looking at the blood supply to the kidneys. So what you need to realize is that the arterial system and the venous system of the kidneys is more like a mirror image. So once you know the arterial system of branches, then it will be very easy for you to remember the venous system. So I think I've already explained that, unless if there are questions that you still don't understand. But here it is. That's why I'm saying it's more like a mirror image of each other. So once you know the arterial system or branches, then it will be very easy for you to remember the venous branches. So it's starting from the aorta, it's branching off to give the renal arteries, the left and the right renal arteries. That will divide to form the segmental arteries. The segmental arteries, they're going to divide to form the interlobar arteries. The interlobar arteries, they can communicate with one another via the arcuate arteries. The arcuate arteries, they will give rise to cortical radiate arteries, and these cortical radiate arteries, they are the same interlobular arteries. That will divide to form the afferent arterioles. The afferent arterioles in the glomerulus, they're going to divide to form the glomerular capillaries. The glomerular capillaries will join to continue as efferent arterioles. The efferent arterioles will form the peritubular capillaries. The peritubular capillaries, they can join to form the cortical radiate vein, which are called the interlobular vein. That can join to form the arcuate vein, and the arcuate vein will join to form the interlobar vein. The interlobar vein will join to form the renal vein. The renal vein will join to join the inferior vena cava, and the inferior vena cava, of course, it will now transport blood to the right side of the heart from the right atria into the left, into the right ventricle, and then it will be pumped into the pulmonary circulation for oxygenation. Then the cycle repeats itself like that. So this is more of anatomy, but I think it's not something that is complicated. So if a question comes to say, can you describe the blood supply to the kidneys? I don't expect you just to write to say you have renal blood flow or renal arteries. The renal arteries are the ones that are supplying blood. So you need to explain the branches of that renal artery. Where is it coming from? It's branching from the aorta and then all the way down. Then the venous, again, you explain. Okay? So we proceed. So let's look at the magnitude of renal blood flow, the renal blood flow. So you realize to say that in as much as the kidneys are small, but the blood supply to the kidneys, it's about 22%, 22 or 20% of the cardiac output. So 22 or 20% of the cardiac output is going to the kidneys, and these kidneys are small. So the increase in blood supply to the kidneys is not necessarily to necessitate the transportation or to necessitate the transportation of oxygen and nutrients to the kidneys. No. In as much as the kidney cells, they also need oxygen, they also need nutrients. But the major function why the blood is being transported to the kidneys is for filtration. So it has got an extra function. Because of that, you need much of this blood to be transported to the kidneys for filtration. That's why 22% of the cardiac output goes to the kidneys. So if you do 22% of the cardiac output, you know to say that the cardiac output on average is 5 liters per minute. 5 liters per minute, that is the cardiac output. So 22% of that 5 liter is going to the kidneys. If you calculate 22% of 5 liter, it will give you about 1.2, 1.3 liters that is introduced to the kidneys per minute. So the kidneys is one of the organs that receives more blood as compared to the other organs in the body, be it the brain or what. The kidneys, they receive a lot of blood for filtration. So the major reason why you have an increase in blood supply to the kidneys mainly is to rapid removal of waste products. So there are waste products that needs to be removed from the blood, and also filtration of body fluids by the kidneys many times a day. So you know to say that the plasma volume is about 5 liter. Plasma volume on average is about, 3 liters, not 5 liters. I think I'm getting tired now. So the plasma volume is about 3 liters. The glomerular filtration rate is about 180 liters per day. If you convert it into minutes, you're going to get 125 milliliters per minute. So in every minute, the kidneys, they are filtering about 125 milliliters, or mils in short. So if the kidneys, they are filtering about 125 mils per minute, or 180 liters per day. If you divide 180 divided by 3, you are going to get 60. So in a day, the plasma is going to be filtered 60 times. So you can see the function of the kidneys. They have to filter the blood plasma 60 times every day. That's huge. So if the kidneys are not operating very well, for instance, in terms of reabsorption, because about 99% of the fluids that are being filtered, they need to be reabsorbed. So if there is a disease that is affecting the kidneys, that is compromising the reabsorption of the fluids, you find that the patient can be dehydrated so fast. Because if you just assume to say there is no reabsorption taking place within the kidneys, it will only take 24 minutes for someone to lose all the plasma. So within 24 minutes, you'll have excreted all your plasma. You will be out of the fluid. So that's why reabsorption is very important within the kidneys, so that you reabsorb much of the fluids. Otherwise, if there's no reabsorption, then dehydration will come in. Okay, so this diagram is just showing the kidney nephron and different components of the kidney nephron and the pressure that is facilitating reabsorption. So as blood is coming in via the interlobular arteries or the cortical radiant arteries, the pressure at which the blood is moving is 100 millimeter of mercury. When it goes into the afferent arterioles, so they will divide, there's a bit of resistance there, then the blood pressure is going to reduce to 60 millimeter per mercury when it's approaching the glomerulus. So in the glomerulus, the blood pressure there in these capillaries is about 60 millimeter of mercury. So this pressure is going to facilitate filtration. In the proximal convoluted tubule, the pressure there of the fluid that is moving is 18 millimeter of mercury. So it means that filtration here to be more because the pressure in the proximal convoluted tubule is less as compared to the pressure in the capillaries. That will facilitate filtration. As you are going to the peritubular capillaries, you can see that the pressure is still reducing. So in the peritubular capillaries, the pressure has reduced from 18 millimeter of mercury to about 13 millimeter of mercury. In the peritubular capillaries, you need the pressure to reduce so much. Why? It's because this pressure can oppose the reabsorption of substances. So the decrease in the pressure in the peritubular capillaries will facilitate reabsorption because the pressure now in the tubes will be more or greater than the pressure in the capillaries, the peritubular capillaries. That will facilitate reabsorption. So you can see that is happening there. So there are changes in the blood pressures as you are moving down the blood supply and also as you are moving down the tubular component of the kidney nephron. Okay. Okay, so I've already explained that. This table is just summarizing the comparison between different types of, a comparison among different tissues of the body in the blood supply, and whether that blood supply is just required for oxygen or is required for other functions. So you can see in the kidneys, the blood flow there in mils per minute per gram of tissue is about 4. So 4 mils or 4 millimeters per minute per gram of tissue. So this is very high when you compare to the other tissue. Like the heart itself, it will just need 0.8, the brain tissue 0.5 milliliters per minute per gram of tissue, the skeletal muscle cell 0.05. But when you're exercising, the skeletal muscle cell blood supply will increase to about 1 milliliter per minute per gram tissue. So the function here, when you look at the difference in arterial and venous oxygen content, so the atrial venous oxygen difference, you can see in the kidneys it's not so much, it's between 12 to 15. So it simply tells you to say that the kidneys, they require more blood supply, but this blood supply is not necessarily required because there is oxygen that is needed by the tissues. It's because of the function of the kidneys. But in the heart there, if you look at the arterial venous oxygen difference, in the heart is about 96, meaning that the blood supply to the heart mainly is required for oxygen supply because it's being used there. Even in the brain, it's the same. Skeletal muscles is also the same. So just note to say that the blood that is supplied to the kidneys is not just required for oxygenation of those cells, but mainly is required for filtration, the cleansing of blood, in other words. So the cleansing of blood from the waste products. Okay. So the functions of the kidneys, the filtration, reabsorption, and secretion. I think I've explained this. But now let's look at the stirring forces that are involved in filtration. The person who asked the question, what do you understand by the word shock?
It's a reduction in cardiac output when there's severe reduction in cardiac output.
Okay.
And
so of course the reduction in cardiac output will result into ischemia or infarction. That explains the reduction in oxygen supply. But basically, when you're looking at shock itself, there are different things that can cause shock. So it could be infections, it could be like bacterial infections or viral infection. So there is what we call circulatory shock. In circulatory shock is due to toxemia. So if there are certain toxins that are being produced by the bacteria, those toxins, they will cause an increase in the permeability of blood vessels. That's where shocks comes in. So the toxins they will go to the capillaries and increase the permeability of the capillaries, so much that the fluids that are confined within the cardiovascular system will now move to the tissues. That will bring about edema later on. But that shift of fluid from the cardiovascular system into the tissues is what will bring about shock, because the plasma volume will reduce so much, the blood volume will also reduce so much, the cardiac output will reduce so much, the blood supply to the vital organs will also reduce. So you find that these vital organs will start shutting down. If they don't have that oxygen, they don't have enough blood supply to them, they will start shutting down. So that is shock. So this shock will have an effect on the kidney itself. Remember, I told you to say they are what we call autoregulatory mechanism of the kidneys. The autoregulatory mechanism of the kidneys, they are there to maintain the glomerular filtration rate despite changes in blood pressure. During shock, there will be decrease in blood pressure, which is called hypotension. So when blood pressure is decreasing, you expect that the glomerular filtration rate has to reduce because the oncotic pressure is going to increase, the hydrostatic pressure is going to decrease. I'll explain those starting forces, you understand. So if there is shock, the blood volume is reducing, there is hypotension, the glomerular filtration rate is going to reduce. But the filtering of blood is very important. So there are autoregulatory mechanisms that will try to maintain the glomerular filtration rate by causing dilatation of the afferent arterioles. So those smooth muscle cells will be stimulated to relax, so they won't be contracting, so that much blood is also going to the kidneys to maintain the glomerular filtration rate. Just get what I'm saying. At the same time, there will be constriction of the efferent arterioles. That is going to encourage a buildup of hydrostatic pressure within the glomerular capillaries that are involved in filtration. But remember that during shock, you want to preserve the blood supply to the brain, because if the blood supply to the brain is reducing, it can also bring issues to do about syncope or fainting, or sometimes stroke. So you can also have a stroke because of the shock. The shock can become secondary to, the stroke can become secondary to a shock. So the body will try now to preserve blood supply to the vital organs, especially the brain. So you find that there will be conflicting information. The autoregulatory mechanism would want to maintain certain amount of blood to the kidneys, but there is the nervous system that will come in, the autonomic nervous system. Because now the patient is under shock, there is stimulation of the sympathetic nervous system trying to resuscitate the function of the heart by pumping much blood to the brain, because the brain depends only on oxygen and glucose. So it will metabolize oxygen and glucose to generate energy. So you need constant supply of oxygen and glucose. So the body will try to maintain blood supply to the brain. So what will happen is that the sympathetic part of autonomic nervous system is going to be stimulated. That will bring about vasoconstriction of the blood vessels that are supplying the kidneys and the muscles, so that you minimize the blood supply to the muscles and to the kidneys. Much of the blood has to go to the brain. So during shock, it depends at what stage of shock you are looking at. So it could be that there are compensatory mechanisms that are inhibiting blood supply to the kidneys. In that way, the glomerular filtration rate is going to decrease because you want to preserve the blood to the brain. It might get complex and complicated because these are a lot of systems that are at play to try and maintain the normal physiology of the body. So they are compensatory mechanisms. But the truth of the matter is that the fluids are moving from the cardiovascular system to the tissues in shock. I hope I've answered that question.
Yes.
Does it make sense, or I've just confused you more?
That makes sense.
All right, okay. Yeah. So there are different stages of shock. Sometimes it can be reversible, sometimes it becomes irreversible. So it depends at what point. Once shocks becomes irreversible, you can't resuscitate the patient. You can't intervene. Whatever you do, you can give drugs, what? The overall result there to be just death. Once the shock becomes irreversible. So before it becomes irreversible, you need to intervene by giving different types of drugs that can help and doing whatever you can. You give IV fluids and stuff like that. Okay, so there was a hand. Someone was trying to ask something. I don't know. Who was that one who was raising a hand? I think it was Eunice Mwanza. Do you want to ask? Eunice Mwanza, are you getting what I'm saying?
No, sir. You can continue.
All right, okay. So I think the last thing that I want to discuss with you are the forces that are facilitating filtration. So I'll quickly go there. So, but before we discuss the forces that are involved in filtration, there is a filtration membrane that is involved in filtration. So the filtration membrane, so this is a capillary, the glomerular capillary, and you have, I've told you to say this membrane is composed of three things, meaning the endothelial cells or the capillaries. In between them, you have fenestrations or spaces or opening. So you can see you have these fenestrations. Then you have the basement membrane after the endothelial cells. Then you have the podocytes. The podocytes, they have these foot-like processes. In between them, there are also openings, which are called filtration slits. So you can see the filtration slits. So if something needs to be filtered, it has to pass through the fenestrations via the basement membrane into the filtration slits. They need to be in the Bowman's capsule or Bowman's space. They need to go into the proximal convoluted tubule. So this is the layer of the filtration membrane. Okay. So these foot-like processes are called pedicels. The foot-like processes of podocytes, they are called pedicels. It's basically the same information. So let's just go to filtration, but mainly I'm interested in studying forces. The forces that are involved in filtration, the hydrostatic force and the oncotic force or pressure. So I hope you are able to see the diagram that I've projected. I want to explain the ultrafiltration pressure or the filtration pressure that is going to facilitate filtration so that there is formation of ultrafiltrate. Blood is brought in by the afferent arteriole. When you check the diameter of the afferent arteriole is larger as compared to the efferent arteriole. You can see that from this diagram. So much blood is introduced as the blood that is leaving. If you compare the two, the blood that is brought in is more as compared to the blood that is leaving the glomerulus, because part of the fluids are being filtered here. So when blood is brought in, the liquid part of blood is called plasma. And in plasma, you have solutes that are dissolved in there. So because more blood is brought in as compared to the blood that is leaving, there is a buildup of fluids within the glomerular capillaries. That buildup of fluids within the glomerular capillaries will create hydrostatic pressure. The hydrostatic pressure is the pressure that is created due to the presence of water molecules. So as the water molecules are accumulating within the glomerular capillaries, they will create hydrostatic pressure that will start pushing the fluids into the Bowman space across the filtration membrane. So across the endothelial cells, the basement membrane, and the podocytes into the Bowman space. So that pressure that is found within the capillary, the glomerular capillary, they are called hydrostatic pressure. So in this diagram, the hydrostatic pressure in the glomerular capillaries is shown as 55 millimeter of mercury. Pay attention and note that the hydrostatic pressure that I'm explaining here is not the blood pressure. We said the blood pressure is coming from the interlobular arteries is 100 millimeter of mercury. By the time it's going to the afferent is 60. But the hydrostatic pressure here is due to the presence of water molecules. The blood pressure can also enhance the hydrostatic pressure because the blood pressure is the one that is introducing the fluids to the glomerulus. The hydrostatic pressure in the capillaries is about 55 millimeter of mercury. Then you have the proteins, the plasma proteins. The plasma proteins, they are so big, and because they are negatively charged, the filtration membrane is also negatively charged. It's very difficult for plasma proteins to be filtered. Under normal circumstances, these big, huge proteins, they're not being filtered. So the concentration of proteins in the Bowman space is almost zero. Almost zero. But you have plasma proteins in the capillaries. So the presence of those plasma proteins, they will create a pressure which is called oncotic pressure or choroidal pressure. So the choroidal pressure or oncotic pressure is one and the same. Is due to the presence of plasma proteins. You know, to say most of these plasma proteins are phosphorylated, so they are carrying a negative charge. And at the same time, the amino group can lose the hydrogen ion, so they become negatively charged. So most of these plasma proteins are negatively charged. Because they are negatively charged, they can attract water molecules towards themselves. So the oncotic pressure or the proteins that are found within the glomerular capillaries, it's going to be an opposing force to the hydrostatic pressure. So it's opposing because it would want to encourage the movement of fluids from the Bowman space back to the capillaries. So this pressure here, it's about 30 millimeter of mercury. So you can see that's the oncotic pressure. It's the opposing force. So it's going into the different direction. That's the oncotic pressure. But you need to know to say that when the fluids are moving into the Bowman space, they will start building up within the Bowman space, and then they'll also create hydrostatic pressure. The hydrostatic pressure in the Bowman space would want to move fluids back to the capillaries. So it's also the opposing force. So you can see here that the net filtration pressure, if you subtract 55 minus 30 minus 15, you're going to get 10 millimeter of mercury. This is called the net filtration pressure. That will now push the fluids from the capillaries into the Bowman space via the filtration membrane. Down here, there's an equation. So you have the hydrostatic pressure in the capillaries minus the oncotic pressure in the capillaries minus the hydrostatic pressure in the Bowman space. It will give you the net filtration pressure. So 55 minus 30 minus 15, you have 10. So these are called stirring forces. The oncotic pressure in the Bowman space is almost zero. But that oncotic pressure, it's going to encourage filtration because you're talking of the oncotic pressure in the Bowman space. So the proteins in the Bowman space, they would want to attract water molecules from the capillaries into the Bowman space. That will facilitate filtration but because the concentration of these proteins within the Bowman space is almost zero, so that oncotic pressure is almost zero. It's not considered. Unless if there is a disease, like in case of proteinuria, whereby you have proteins in the urine. So due to maybe damage to the membrane that is involved in filtration, maybe it could be an autoimmune disease that is attacking the membrane. It could be inflammation that is taking place there. In chronic renal diseases or acute renal diseases, then you have movement of proteins into the Bowman space. You find that the oncotic pressure there is going to increase. So when you have proteinuria, even the volume of the urine is going to increase, because that is also going to facilitate a filtration of fluids. So proteinuria with polyuria. So you're going to have proteinuria with polyuria. Okay, just like you can have glucosuria with polyuria in diabetes. So I'll explain all that later on. But I hope you are relating. Okay, so those are the forces that I was explaining: the colloidal pressure, the oncotic pressure, the hydrostatic pressure. So this is just your information to go and read on, but otherwise, most of this information I've already explained. Okay, so with that information, I think for today, it's enough. Next time we meet, we'll look at the glomerular filtration rate and the factors affecting it. So the filtration membrane, you already know it by now. If I ask you to draw it, you are able to draw the endothelial cells, the basement membrane, and the podocytes. The pedicell, which are the food processes of the podocytes. So you have the filtration slits there in the endothelial cells between